Greetings! Welcome to the lecture of Plant Molecular Biology on plant tissue culture. In this lecture, I'm going to discuss with you about aseptic techniques and principles of media design. I'm Wilson Yong from the Biotechnology Research Institute, University Malaysia Sabah. Plant tissue culture broadly refers to the in vitro cultivation of all plant parts under aseptic conditions on a nutrient culture medium of known composition. The excised piece of tissue or organ obtained from a plant to be cultured is called an explant. The explants can be taken from many different parts, including portions of leaf, stems, roots, cotyledons, embryos, and many types of matured cells. However, the selection of an explant largely depends on the purpose of doing tissue culture, suitability of an explant for initiating cultures, and availability of an explant. While many different explants can be used for tissue culture, axillary buds and meristems are most commonly used, especially for micropropagation, because they are less prone to genetic changes than disorganized tissue. Other desirable properties of explants include easily sterilizable, juvenile, and responsive to culture. The success of the plant tissue culture depends on the choice of suitable growth medium. Tissue culture media should generally contain energy sources or macronutrients and inorganic salts or micronutrients to support plant cell growth. The media can be in the form of liquid or semi-solid, depends on the types of culture. Other requirements for plant tissue culture include growth regulators, biofertilizers and vitamins. External parameters such as light, temperature and pH, and subculturing frequency also play roles in successful tissue culture by supporting plant growth and tissue development. Aseptic technique is essential for the successful establishment and maintenance of plant cell, tissue, and organ cultures. The in vitro environment in which the plant material is grown is also ideal for the proliferation of microorganisms. In most cases, the microorganisms outgrow the plant tissues, resulting in their death. Contamination can also spread from culture to culture. The purpose of sterilization or aseptic technique is to eliminate the contaminants or minimize the possibility that microorganisms enter or remain in the cultures. The list of organisms described as contaminants in plant tissue cultures includes bacteria, fungi, viruses, yeast, and mites. Contamination with bacteria is thought to be the most serious and has been described extensively in the literature. Bacteria are usually introduced with the explant and may survive surface sterilization if embedded in the interior tissues. Some bacterial spores can also survive the sterilization procedure even if they are on the tissue surface. Many kinds of bacteria have been found in plant tissue cultures, and they can be recognized by a characteristic ooze with many different colors including white, cream, pink, and yellow. Fungi may enter cultures on explants as plant pathogens. They are frequently present in soil, or spores may be airborne. Fungi may be recognized by their fuzzy appearance and occur in a multitude of colors. Next contaminant is virus. Viruses are extremely small that are not easily detected Thus, plant culture is not necessarily pathogen-free, even if microorganisms are not detected, and viral contaminant can influence culture success. Contamination in tissue culture can originate from two sources, either through carryover of microorganisms on the surface or in the tissues of explants, or through faulty procedures in the laboratory and tissue culture technique. Most contamination is introduced with the explant because of inadequate sterilization or just very dirty material harvested from the field. Initial contamination is obvious within a few days after cultures are initiated. Latent contamination is usually by bacteria and is often observed long after cultures are initiated. Apparently, the bacteria are present endogenously in the initial plant material, but not obviously pathogenic in situ. Once in vitro, however, they increase in titer and overrun the cultures. Latent contamination is particularly dangerous because it can easily be transferred among cultures. Contamination can also occur as a result of poor sterile technique or dirty lab conditions. This kind of introduced contamination is largely preventable with proper care. Whether it is labware or culture medium, plant material or instruments used for culture in the laboratory, all are sources of infection and need to be sterilized. The routine sterilization procedure of non-living articles, such as nutrient media, glasswares, distilled water, and instruments, is by autoclaving under steam at a pressure of 15 pounds per square inch in a temperature of 121 degrees Celsius for 15 minutes. It should be noted that when autoclaving screw cap bottles, the caps must not be closed too tightly in the autoclave so that gases can expand without the risk of explosion. An alternative method of sterilizing glass goods and instruments is by heating in an oven at 150 degrees Celsius for 3 to 4 hours. Thermolabile compounds are often sterilized either at room temperature or in cold by passing through bacteria filter. Plastic containers such as petri dishes or plastic centrifuge tubes that cannot be heated are usually sold after sterilization with ethylene oxide gas, and they cannot be re-sterilized. Items in the transfer hood or laminar flow may be sterilized by germicidal lamps. UV lamps should not be used when people are present because the light is damaging to eyes and skin. Plants left under UV lamps will die. It is also possible to sterilize items in a microwave where necessary. Plant material, which is to be cultured, should be surface sterilized to remove the surface-borne microorganisms. This procedure is done in front of a laminar air flow or inside the inoculation chamber before the plant material is placed onto the culture medium. Commonly used disinfectants for plant tissue culture include calcium hypochlorite, sodium hypochlorite, hydrogen peroxide, and others. The use of two-step sterilization procedure, which involve placing the explants in a 70% ethyl alcohol solution prior to treatment with another disinfectant solution, has proven beneficial to explant sterilization. For one of the examples, commercial bleach contains about 5% sodium hypochlorite, so the use of concentration at 10 to 20% is equivalent to 0.5 to 1% of sodium hypochlorite. Adding a wetting agent such as Twin 20 or Twin 80 into the disinfectants can reduce surface tension and allow better surface contact between the plant tissues and the disinfectants. Conducting the sterilization process under vacuum can result in the removal of air bubbles and provides a more efficient sterilization process. The procedures of aseptic technique during plant tissue culture include the following. Put on the switch of UV lamp of inoculation chamber for 1 hour before work, and remember to turn off the UV lamp before entering inside or work in the chamber. Exposure to UV light builds up a high concentration of ozone gas, which is toxic, inside the closed chamber. It is therefore healthy to enter the chamber only 15 to 30 minutes after switching off the UV lamp. Switch on the power of laminar airflow to the air blow for at least 15 minutes before work. Put all the sterilized articles such as media, instruments, and glass goods on the glass racks of the inoculation chamber or the table of airflow cabinet, and the airflow will blow the bacteria-free air over the working surface. The working glass tabletop of the inoculation chamber or the table of laminar airflow is swabbed with alcohol before starting work. Pour alcohol in a clean coupling jar and dip all instruments into it. After that, flame the instruments using Bunsen burner or spirit lamp each time before use. Always wear a clean lab coat and use a mask. Clean the hands with alcohol and dry it. Always keep away the hands moistened with alcohol from the Bunsen burner. Take the surfaced sterilized or aseptic plant material in a sterile petri dish. Flame the neck of culture tube or flask and quickly remove the plug of glass vials before transfer the tissue onto the medium and replace the closure. Lastly, do not dip hot instruments in alcohol and do not use hot instrument for cutting or holding the plant material. Nutrient medium for plant tissue culture is a mixture of substances on or in which plant cells, tissues, or organs can grow and survive. These substances include salt mixture, which can be classified into macronutrients and micronutrients according to their relative concentrations in plant tissue. Organic substances such as sugars, vitamins, and amino acids. Natural complexes comprising growth-promoting or inhibitory compounds. Inert supportive materials, and most of the time plant growth regulators to control different physiological responses in vitro and direct the morphogenesis of plant tissues in culture. Plant tissue culture media provide water, minerals or nutritional needs, vitamins, growth regulators or hormones, and other desirable nutrients including various organic compounds for the growth and development of plant cells and tissues. The media also provide access to the atmosphere for gaseous exchange and act as a dumping ground for secondary metabolite waste. The inorganic nutrients in culture media consist of a wide range of mineral salts. Nutrients needed in large quantities or more than 0.5 millimole per liter are macronutrients, and those that are needed in very small amount or less than 0.5 millimole per liter are called micronutrients. Different types of media, their composition, and different roles of their prominent elements can be found in literature, and the success of a plant tissue culture depends on the choice of suitable nutrient medium. Basic media that are frequently used for plant cell and tissue culture include MS or Burasich and Scoop medium and Gamborg P5 medium. Other formulated media such as NN medium, SH medium, and WPM medium are also available for different culture systems. Different plant systems have different nutritional requirements, and therefore there is no single medium which can be used for tissue culture for all the plants. The 6 elements, namely nitrogen, phosphorus, potassium, calcium, magnesium, and sulfur, are the essential macronutrients for tissue culture. They are required in relatively large quantities and play an important role in plant growth and development. Their functions range from being structural units to redox-sensitive agents. Generally, application of macronutrient increases yield, growth, and quality of plants. Nitrogen influences the rate of plant growth and is present in the largest percentage of all the essential elements. It is an essential element in the molecular makeup of nucleic acids, proteins, chlorophyll, amino acids, and some plant hormones. Nitrogen deficiency is normally characterized by chlorotic leaf and stunted growth. Excess nitrogen promotes vigorous growth but can suppress flower and fruit development. Common sources of nitrogen for tissue culture media are ammonium and nitrate compounds. Phosphorus is abundant in meristematic and other fast-growing tissues. As part of DNA and adenosine triphosphate or ATP molecules, phosphorus is an essential element in photosynthesis and respiration, and it affects plant maturation and root growth. Stunted growth and abnormal discoloration can be symptoms of phosphorus deficiency. Potassium phosphate and sodium phosphate are routinely included in plant tissue culture media as a source of phosphorus. Potassium is necessary for normal cell division and promotes meristematic growth. It plays a role in many reactions within plants, such as synthesizing carbohydrates and proteins, and manufacturing chlorophyll. Insufficient potassium results in weak and abnormal plants, sometimes with necrotic margins on older leaves. Potassium nitrate and potassium phosphate are common sources of potassium in culture media. Potassium chloride is also occasionally used. Calcium is an integral component of plant cell walls, where it plays a role in the formation of pectin, a substance that bonds cell walls together. It assists in growth and promotes root and leaf development. It also helps to control permeability and facilitates the movement of carbohydrates and amino acids throughout the plant. As calcium oxalate, it ties up oxalic acid, which is a toxic byproduct of protein metabolism. Distorted or dead shoot or root tips can be a sign of a lack of calcium. Calcium is usually included in tissue culture media as calcium chloride or as calcium nitrate. Calcium phosphate is also sometimes used. Magnesium is the central element in chlorophyll molecules. It is also important as an enzyme activator. Magnesium deficiency causes pale and sickly foliage. Most tissue culture formulas call for magnesium sulfate, or commonly known as Epsom salts, as a source of magnesium. Sulfur is present in some amino acids and enzymes. It promotes the deep green foliage and root development. A deficiency results in overall yellowing of the plant. Sulfur is supplied in tissue culture media in conjunction with other elements as various sulfate compounds. In addition to the major elements that plants require, a number of other elements are essential to good growth but are needed only in extremely small quantities. They are called minor elements, trace elements, or micronutrients. Several micronutrients are toxic to plants in excess amounts. Few of the important micronutrients will be discussed here. Iron is involved in chlorophyll synthesis. It also participates in energy conversion in photosynthesis and respiration, as it is reduced from the ferric or trivalent to the ferrous or divalent state. In tissue culture media, ferrous sulfate is often mixed with the sodium salt of EDTA to sequester the iron, making it more readily available to the plants. Boron is an important micronutrient presumed to play a role in the movement of sugar, water, and hormones. It is also involved in nitrogen metabolism, fruiting, and cell division. Lack of boron produces deficiency symptoms such as tip dieback and chlorotic leaf. However, excessive amounts of boron can cause plant injury or death. Boron is added to tissue culture media in small amounts as boric acid. Cobalt plays a critical role in the overall growth process of plants. Cobalt is necessary for the processes of stem growth, elongating the coleoptiles and expanding leaf disc. It is a critical element needed for a plant to reach maturity and for healthy bud development. Other micronutrients include copper, manganese, molybdenum, and zinc. As a plant nutrient, copper seems to function primarily as a cofactor for a variety of oxidative enzymes. Copper is used in the form of copper sulfate in culture media. Higher concentration may be toxic. As a cofactor, manganese is required by some enzyme reactions, particularly in photosynthesis, respiration, and nitrogen assimilation. Manganese is also involved in pollen germination, pollen tube growth, and root cell elongation. Usually, manganese is added to the medium in the form of manganese sulfate. Molybdenum is a cofactor in two enzymes that convert nitrate into nitrite, a toxic form of nitrogen, and then into ammonia before it is used to synthesize amino acids within the plant. It is supplied as sodium molybdate in media. Zinc is an activator of enzymes that are responsible for the synthesis of certain proteins and is used in the formation of chlorophyll and some carbohydrates and conversion of starch to sugars. Zinc is essential in the formation of auxins, which helped with growth regulation and stem elongation. Typical symptoms of zinc deficiency include shortened internodes and smaller leaf. Zinc sulfate is the most common form added to culture media. Organic substances are compounds containing carbon, such as carbohydrates, proteins, hormones, enzymes, and so on. They are not usually provided to plants because plants normally make their own food. However, plants in culture are too small or incomplete to synthesize all the organic chemicals they need. So, organic substances must be added to the tissue culture media to augment the plantlet's atmospheric or self-generated supply. Sucrose is a very important part of nutrient medium as an energy source, since most plant cultures are unable to photosynthesize effectively in tissue culture vessels. Sucrose consists of two chemically bound monosaccharides, fructose and glucose, and is an indirect product of photosynthesis. A high concentration of sucrose, generally 30 grams per liter or 3%, is suggested for most media formulas. Occasionally, fructose or glucose is also substituted for sugar in media. Vitamins are organic substances required for metabolic processes as cofactors or parts of enzymes. Hence, for optimum growth, medium should be supplemented with vitamins. Adenine or vitamin B4 is important to cell as part of the nuclear substances, DNA and RNA. It is used in culture media as adenine sulfate to promote shoot formation. Myoinositol, a sugar alcohol in the B complex, is required in many media. In its phosphate form, it is part of various membranes, particularly those of organelles such as chloroplasts. Though not essential to growth, it is often beneficial in stimulating cell growth when added to tissue culture media. Thiamine is essential for most culture media because it functions as a coenzyme to assist the organic acid cycle of respiration, which known as citric acid cycle or Krebs cycle. Amino acids are building blocks of proteins, some of which combine with nucleic acids to form nucleoproteins. These nitrogenous substances are not essential, but addition of amino acids augments the nitrogen supply to media, which is important for specific cases like stimulating cell growth in protoplast cultures and also in inducing and maintaining somatic embryogenesis. This reduced organic nitrogen is more readily taken up by plants than the inorganic nitrogen. L-cysteine, L-glycine, L-glutamine and L-asparagine are commonly used amino acids that are being added to the culture medium in forms of mixtures, as individually they may inhibit cell growth. A natural complex can be used when a defined medium fails to support a particular growth response, and many additions serve various purposes. Natural complexes added to the medium generally make the medium undefined, since variations in growth-promoting or inhibitory compounds in these complexes are to be expected. Some examples of these natural complexes are coconut endosperm, yeast extract, malt extract, tomato juice, banana, potato extract, casein hydrolysate, and fish emulsion. Antioxidants are sometimes used if there is excessive browning of the explant, and they retard oxidation of the explant. Examples of antioxidants are citric acid, ascorbic acid, pyrogalol, and frologlucinol. Adsorbents are also used to promote morphogenesis by absorbing inhibitory compounds in the culture medium and substantially decreasing the toxic metabolites, phenolic exudation, and brown exudate accumulation. Two adsorbents are polyvinylpyrrolidone or PVP and activated charcoal. Activated charcoal release substances naturally present in the charcoal that may promote plant growth. It also absorbs vitamins and plant growth regulators from the medium and gradually release them to the explant. Many tissue culture experiments are conducted on some types of stationary support such as filter paper, cotton, cheesecloth, vermiculite, and special membrane broth with a liquid medium. However, gelling agent is the most commonly used supportive material. The type of gelling agent used can influence the type of growth response in the culture. If the gelling agent is unwashed or not purified, it will generally discolor the medium because it contains various impurities. Sometimes dramatic differences in explant response can be observed by changing the brand of solidifying agent used. Three types of gelling agents, namely agar, gel-rite, and gellan gum, are mainly used. Agar is a mixture of polysaccharides derived from extracts of several species of red algae or seaweed, and can easily bind with water to enable explant to be slightly embedded on culture media and allow nutrients to diffuse through the medium to the plantlets. Gel-rite and gellan gum are transparent in appearance and are polysaccharide complex produced as a fermentation product from a Pseudomonas species. These gelling agents form clear gels, unlike agar, which is translucent, and hence it is easier to detect contamination, which might develop during culture growth. Their prices are twice that of purified agar, but only less than half as much gel-rite or gellan gum is necessary to provide the same gel strength. Plant growth regulators are phytohormones that are used to order the growth of a plant or plant part. Many are endogenous hormones that are produced naturally in plants, but plants that are growing in vitro usually do not manufacture sufficient quantities of growth regulators, so they must be added selectively to culture media. Auxins, cytokinins, gibberellins, abscisic acid, and ethylene are the substances used as hormones or growth regulators. The auxins and cytokinins are used most frequently in tissue culture media. In general, auxins promote cell enrichment and root initiation, whereas cytokinins promote cell division and shoot initiation. An auxin is required by most plant cells for division and root initiation. Auxins commonly used in plant tissue culture media include indole 3-acetic acid IAA, indole 3-butyric acid IBA, naphthalene acetic acid NAA, and 2,4-dichlorophenoxyacetic acid or 2,4-D. The auxin 2,4-D is widely used for callus induction, while IAA, IBA, and NAA are used for root induction. However, at high concentrations, auxin can suppress morphogenesis. Auxins differ in their physiological activity and also to the extent that they are bound to cells, metabolize, or move through tissues. Cytokinins are derivative of adenine, promote cell division and stimulate initiation and growth of shoots in vitro. The frequently used cytokinins are 6-benzylaminopurine or BAP, 2-isopentenyl adenine or 2-IP, kinetin and zeatin. They modify apical dominance by promoting axillary shoot formation. A high concentration of cytokinin causes inhibition of root formation and promotes adventitious shoot formation. The ratio of auxin to cytokinin plays an important role in morphogenesis. A high ratio leads to embryogenesis and root initiation, whereas a low ratio leads to axillary bud and shoot proliferation, though an intermediate ratio is required for the formation of callus. For morphogenesis, many plant tissues have an absolute requirement for a specific cytokinins for morphogenesis to occur, whereas some tissues are considered to be cytokinin independent. Gibberellins are plant hormones that regulate various developmental processes, including stem elongation, germination, dormancy, flowering, flower development, and leaf and fruit senescence. They are mainly used to induce plantlet formation from adventive embryos formed in culture. Abscisic acid is used in plant tissue culture to promote distinct developmental pathways such as somatic embryogenesis and for culturing woody species. It inhibits cell division and has a favorable effect on abscission. Ethylene is associated with controlling fruit ripening in climacteric fruits, and its use in plant tissue culture is not widespread. Some plant cell cultures produce ethylene which, if it builds up sufficiently, can inhibit the growth and development of the culture. In conclusion, the preparation of culture media and the underlying cultural technique are inextricably related and have an impact on each other. Even if the cultural technique is perfect, the quality of the culture media used could still affect plant growth and development if it was not properly prepared. That is, if it was not sterilized or if the experimenter made a mistake during the preparation process. On the other hand, if the cultural technique is not properly executed, the consistency of plant tissue culture would be affected. With that, I would like to end this presentation. I hope you enjoyed the topic. Thank you so much for joining me and participating in this lecture. Thank you.